Okay. So hopefully you guys can all see my screen. As you should know, we're going to be talking about bioassessments today. This will be a little more relevant. I think we have a stream survey in a few weeks. So basically a bioassessment is an evaluation of the biological conditions of a water body. It uses a survey of the resident biota. And these can basically be thought of, so the resident biota can be thought of as kind of continual natural monitors of environmental quality, and they'll respond to both short-term and long-term trends in pollution or habitat alteration. And so these bioassessments will be assessing the survival, reproduction of these biota. So basically kind of just the abundance. So we can see if like there's a low abundance of a particular biota, or if there's some biota missing. Like you remember in the macroinvertebrates lab or the aquatic organisms lab, we covered the feeding guilds. So if there's a feeding guild that's not represented, something like that, then we know it's probably an impaired stream. Although we may not know exactly why it's impaired, and then we'd have to do a little more survey, another type of survey probably, look at water quality or something. But basically the goal is to preserve biotic integrity, or the capacity of a watershed to maintain a balanced community that reflects the natural assemblages of the organisms and functional organization in that region. So basically you'd just be recording a number of variables. So you might be looking at fish, macroinverts, microbes. You might be looking at overall richness, abundance. You might be looking at reproductive success, or toxicology. So, are there pollutants building up in certain organisms, things like that. Bioassessments can cover all of those and more. So I'm not sure if we'll be doing this in the stream survey, but this is a common way to get macroinvertebrate samples from a stream. And basically what you'd do is you'd stand upstream. You have this little square net called a kick net. You stand upstream of it and you just basically kick the rocks or substrate around on the riverbed. Usually you try to do like 1 square meter area. And this will like break up and release those macroinvertebrates that are on the substrate, and they'll flow downstream and get caught in your net. And then you can take that and I guess they'll lay it out in a tray, and you just pick some a few numbers randomly and you subsample it. So you identify the organisms there. So you do about 20% of these. And I'm not sure if we're doing that in the stream survey or not. So this bioassessment, you can have 2 things. You'll have community metrics, which would be like the loss of sensitive taxa. So this could be the total EPT, the total taxa, or the EPT richness. So if you'll remember EPT, I'm not sure if I actually introduced it to you like that, but EPT stands for Ephemeroptera, Plecoptera, Trichoptera. So those are those 3 orders of macroinvertebrates we covered in the aquatic organisms lab, and you'll remember that they're all sensitive to pollutants. And they're very common throughout most watersheds. So that's kind of why they're chosen as an indicator species. And so by looking at those are orders. Ephemeroptera, Plecoptera, Trichoptera are orders of organisms. So you'll be looking at the number of species within each of those orders, and you'll just sum them up, and that'll be your EPT richness. And then the percent EPT will be the percent of EPT organisms to the whole sample, right? So if those are really low or you're missing some entirely, like loss of sensitive taxa, that's an indicator that there's some sort of pollution. On the other hand, you might have an increase in tolerant taxa. So you'll have certain species that are pretty tolerant of pollutants. And if they're increased, you can tell basically like it's just kind of like the flip of this coin, right? So it says percent dominant. A lot of times you look at the percent, like the top 2 dominant, and what the percentage of them are to the overall sample, how much of the overall sample they make up. So these multi-metric indices of biotic integrity, they're sometimes referred to as stream condition indexes, or SCI, and we'll be covering the West Virginia stream condition indice. They rely on characterizing reference conditions that are in the region that you're interested in, and identifying real key biological attributes that respond to changes in stream conditions that you might see. So it incorporates multiple biological and community level characteristics. Yeah, so and that's just basically like the measure of diversity, species composition, the functional feeding group representation, and then the abundance of, so it also includes these tolerant and intolerant species. Sorry, I want to go through this a little quick because I want to leave us enough time for the homework. So the West Virginia Stream Condition Index basically it allows us to identify streams that are stressed by human disturbance or pollution within West Virginia. So this is unique to our state, but it's similar to others. So there was a study done, and they came up with 6 metrics. Over here in this right hand, this is the expected response to disturbance. So total richness, total number of species should decrease if there's some sort of stressor like pollutants or habitat alteration. EPT richness, which I covered, is the number of species that are either Ephemeroptera, Plecoptera, or Trichoptera, will decrease. Percent EPT, so how much of the overall proportion do those make up? That'll decrease if it's disturbed in some way. So percent Chromonidae, they're pretty tolerant. So these are like your lake flies or non-biting midges. They'll increase. Percent of top 2 dominant taxa will increase. And this last one, the Hilsenhoff Biotic Index, it's basically an abundance weighted average of tolerance of the assemblage of organisms. So they have values, I think, between 1 and 10 that represent a particular organism's tolerance of pollutants. 10 being very tolerant, 1 being intolerant. And then they'll use that along with the abundance of the individual organisms to calculate this HBI, the Hilsenhoff Biotic Index. So those are the 6 that we'll be interested in for the West Virginia Stream Condition Index. So then you'll take those 6 and you'll standardize them. And you guys don't have to do this in the lab. It's done for you automatically in the Excel spreadsheet. I just want you to know. It standardizes them so that each of those 6 are out of 100. And then you average them to get an overall score. So you'll only have one score for each site, and that'll be between 0 and 100. And if it's less than 65, it's considered impaired. Greater than 65, it's not impaired. And again, this is just kind of a general rule of thumb. And this is what we use in West Virginia to find an impaired stream. And then some of the conditions that might cause that would be like low DO, low pH, and then some sort of they have habitat scores. So like if habitat's poor, again, that's going to affect this as well. And you're going to see that in the response of those 6 metrics that we saw. So in today's lab, you should have hopefully downloaded. There's an Excel file. It contains some water quality data from the Cheat River watershed. It was done by some West Virginia graduate student probably a while ago. So some of the streams were impacted by acid mine drainage and acid precipitation. It's kind of a result of that. And we'll just be comparing the streams and discussing them. And again, like all those calculations will be covered or pretty much done for you in lab or in the spreadsheet. So just real brief, this is the acid mine drainage overall reaction. So we'll have I guess like iron sulfide, FeS2. Now it'll react with the water and oxygen to produce this basically sulfuric acid. So we're increasing the pH here. So it could result in low pH, even as low as -3. This is kind of a big common problem in West Virginia, because we have a lot of coal mines here, and that water that comes up from those does run off into streams. Although we do have some ways to mitigate that, it doesn't always work 100%. So if the pH increases, so it becomes more basic, often we'll dump calcium carbonate into streams to help mitigate the changes in pH. Then the heavy metals will precipitate out, I guess, around these pHs. But this will cause an increase in conductivity, which can also have some effects for organisms in the river. So that's not exactly the most important thing to know. I just wanted to introduce it to you. Are there any questions before we move on? Is my screen still sharing? Because it seems like I didn't hit the end presentation button. Okay, thank you. So if no one has any questions on anything we covered, we can launch into this spreadsheet. Hang on. So this is what it should look like. So go ahead and load it up. So this is where we'll be basically copying and pasting in here and building our figures. Trying to find everything keeps opening on the wrong monitor. So here's the homework assignment. I'm not going to read through all the objectives. You guys can do that. But basically, here's what it's going to tell you to do. we're going to do it. And then we're going to make these figures. And then these figures, and then there's some discussion questions here at the end. And I think we'll get through it all. So this abundance tab is basically where all of our raw data is. In this tab, the SCI calculations tab is where our analyses will be done. This box is the same as this box. I simply like transposed it because you can just copy and paste these cells like this. Once you paste into here, it'll automatically calculate all these values for you. And you can just copy and paste it into the corresponding site over here. So I'll go through that. Hang on, my monitor blinked out. One thing to really pay attention to, like some of these names here, the stream names are very similar, and there's even 2 muddy creeks. So really pay attention. I would look at the red number here. So when we're copying and pasting this into this column here, the B column, you're going to then take this information down here on the green. Can you guys see my screen turning black like that? We're going to copy this into here on the abundances tab. And just make sure that it's matching with this number here because they don't go in order. So I'm going to work along with you. You can make sure you might be able to just copy the whole column. So I'm just going to take this Beaver Creek column here, scroll up and paste. And you can see it automatically calculated my scores here. So I'm going to copy all those. And I want WVMC12B11, Beaver Creek, WV1B, all that. And then you're just going to paste the values. So go ahead and start doing that. I'll do it along with you. And get all of these filled in. Okay. And let me know if there's any questions. So again, I'm just this time 17-0.7A. Just selecting that whole column, Control V. It automatically updates. Go back to site preference. Let me check. That was Crab Orchard 1707. Yep, this next one, but they're not all in order. Why don't you guys go ahead and do that? I'm gonna see if I can't fix what's going on with my computer.
Yeah, while Alex is doing that, if you guys have any questions, I'd be more than happy to help out as well.
All right, hopefully that fixed it. You guys know I've had trouble with my computer, so I'm on a new computer that I had been trying to build for a while, but the graphics card I bought was really cheap. And so my monitors blink randomly. I don't lose any data, or anything, but I'm not sure if you can see it. And I'm not sure, I can't, certainly. It only blinks for a few seconds. And let us know if you get, okay, thank you, Megan. Let me know if you get done with this and we can move on. Yes, you see his third one, Daughtry Run, is down here. So it's important to make sure you get these in the right row.
I had a quick question.
Yep.
I was wondering if it mattered where we put the muddy creeks, like if one was supposed to go in a certain spot.
Yeah, so it does. So that's why I think they're even bolded here. That's why I said reference those red numbers.
I
So
see.
that's the end code.
Okay.
Yeah, otherwise it'll just be mislabeled. And it'll be harder to discuss it. All sorts of trouble.
I have a question.
Yep.
So when I'm copying the data from abundance and I'm putting it into the WV psi calculations, it's not giving me the same numbers that you're getting.
Okay. You're pulling from these. It's
Yeah.
also possible that I messed up. Let me
Yeah,
open
I'm
up.
pretty sure it's me because it keeps like it's reading the first one as like 83.9, but the ones after that, they're all like the same. They're all 83.9, each one of them.
Is this one correct?
That looks correct, yeah. Maybe if you just redid like the first one.
Yeah, so let me see if I can hang on. Is anyone else having that trouble?
And mine's also not pasting right. It keeps telling me I have errors.
That might be the difference between if you paste just regularly, like Control V.
Oh, yes.
Excel is actually going to try to reference the equations in. So if you need, that's why Alex is just pasting the values, because then it's basically just copying the values that you're getting from these equations and then pasting them. So you can't just do sort of the right or regular paste for this. That might solve your problem. It might not.
Well, I figured that out, but it was still giving me the wrong numbers. Like it was still pasting the incorrect numbers.
Are you pasting the values into column B or column C?
Column B.
Okay.
So if that's not working for you, you can either take these, I think, so if I highlight these, Control C, and then I do that, I think, let's see, no, transpose won't work. So you can Control C, just go anywhere, Control C, paste those values, and then you can copy those. And then in the right place, you hit this one, transpose. Now it should be in the right order. You see, it's like another step. It's annoying. But you can do that. So once you have them in lists, you can't do it. You have to paste it, paste just the values first. So I took this and went over here, right click, paste values. Then you can Control C the values. Just where you would go over here and select whichever one it was. And then you do this one here that says transpose, and it should work. That's one solution. The other solution is to go through the formula and before like each letter put a $sign, and then before each number put a $sign. You have to update all of those. and then you can control C. So I'll just do those. Control C with the formulas, and then transpose with the formulas. Alternatively, I can show you what the formulas should be for each cell if you want to see them. So this EPT, if you just want to type them in, should be equal I33. Richness should be equal I39. Sorry, I should start the percent EPT is I32. EPT is I33. Richness is I39. Percent chiron is O equal O32. So yeah, O32. MHI equal I37. Percent Dom I38. And then this one's gonna be a little harder, but I can walk you through it. So it's going to be equal averages, all capital, and put your parentheses. The first thing is select these two rows. So it's going to be J32 in K32. So it's these two cells. You should just be able to drag, add a comma, and then these three cells, so 37 through 39, and then another comma, and then you want this correlation value over here, so P32. Alicia, so what I'm doing is if someone had a problem where these values were not matching these values and you wanted to update the formulas, you could do that. Alternatively, you can just copy these values, go over anywhere else, paste the values, copy them again, go here, select where you want to put them, and then transpose. So I don't know if anyone else was having that problem. I'm sorry that that happened. Were you the only one who was saying they had that issue?
It was me, but I'm just like manually putting in all of the data now.
You can do that too. It came like this when I was given this lab, and I moved it like this so it would be easier. And I don't know why why it wouldn't work for you. My guess, are you on like Mac or something?
Unfortunately, yes.
Yeah, I'm sorry. It's hard for me to anticipate Mac problems because I don't have one. Well, I have that virtual machine, but I don't have any Excel or anything on it. Yeah. So sorry about that. I'd hoped it would work easier that way. So who all is done with that? Who all has this full site data thing? I don't understand. Hang on. For some reason, it's not giving me the box when I come back here. Okay, Joseph, you're filled in. All right, let me ask this. Is it just Joe that's done? All right, who's not done? Kylie, okay. Luke, okay. I'll give you guys a few more minutes. Let me know when you are. So I'm assuming it's just you three that aren't done. I want to get through making the figures. And I think what I'm going to do is I'll make one from each section. And then I will turn you guys loose. But I'll be here to help you. And if you want to move ahead, and if you're done and you want to move ahead, you feel confident, feel free to. And in fact, actually, let me just do this. For you guys who are done. I'll just show you. If you're not done, keep working and I'll do another little demo on this. But I'm gonna show them how to do one of these figures and then I'll put you guys who are done. I'll assign a breakout room and just let you guys move in there as you're done. So go up to, I like to just pick a random blank cell, go up to insert. Actually, I like to have one way up here. We'll see. Insert, and we want a scatter plot. Just this one will be fine. So it's blank. Go to select data. And the first one we're going to want to do, if you look at question 2, A is conductivity on the Y versus pH on the X. So I'm going to hit this little add button here and this pops up. You don't really have to worry about the series name. So we want, what I did here is I just clicked on X. Now I'm going to, so the X, and I want pH for that. So I'm going to go here to pH. I'm just going to copy this. I'm not going to do this bottom row because it's NA. I'm going to hit enter. Then for the Y, I'm going to click here. It wants conductivity. Again, I'm not going to do the bottom row. Now I'm going to hit OK. And then again, I'm going to hit OK. And I need my axis labels. So I'm just going to put, and then pH down here. And then because I have conductivity and pH, I don't need a chart title. And then I don't like to have these, so I'm just going to delete. So you select these and delete. The next thing we need to do is to add a trend of a best fit line. So I'm going to go down here to trend line, but I'm going to hit this arrow and do more options. So this thing should pop up. You want to click these bars over here. And then you want to set display equation and display R squared. I don't know why it puts it there, but you're gonna have to move it. Again, select the line so that we're working with that line. All right, so I just clicked on this little line. It gives me a linear one by default. And we want to pick the one that's the best fit. So we want the highest R squared value. So I've clicked on this line. You can see that because it has these little dots at the end. So linear is 0.5, and we're going to choose between linear, log, power, or exponential. So linear is 0.5. If I did log, it's 0.6, so that's my better one. If I did power, it's 0.4, so that's not better in log. If I did exponential, it's 0.3, so logarithmic was the best one. So I'm gonna just select logarithmic, and then I'm done with conductivity versus pH. Moving this a little bit over. So that's how you do that first one. And it's going to be the same exact thing for all of the other figures. Okay. So we're going to start working through those. Is anyone not done? Okay, so everyone's done. All right, I'm going to do this. Levi, do you feel comfortable? I'm going to split into 2 groups. I think I have to end my screen share. And Levi, I think you should have screen sharing privileges.
Yeah.
So Levi, you're in room 1, and I'm gonna go to room 2. Hello, guys. So are you able to go to the breakout room? So let me know if you guys are having problems joining your breakout rooms. If you don't join your breakout rooms, I'm going to assume that you're not here paying attention and participating in class, and you will lose participation points. So fair warning.
You're muted, Alex.
Hi, Anna. I know you were gonna be late today. I took note. How long were you waiting? Hey, I can't hear you if you're talking. So we're done. Everyone's left. I can go through if you want and give you the lecture real quick. The video will be uploaded, however. I can give you the little demo too on what we're doing. We have a few minutes if you're interested. Otherwise, you can wait until the video comes out. Let me know what you want to do.
Okay, it looks. She said that she'll just watch the video.
Okay.
In the chat.
Cool. Yeah,
So-
just let me know if you have any questions or anything, and I'll have you down for attendance. Do check that your past grades, like if you have any questions or think something wasn't right, those should all be available. So just check those and yeah, let me know if you have any questions.
Have a good evening.
All right. So yeah, hang on.